You must have heard the term genetic engineering and wondered what it means. Let me show you what it means. Basically, we have talked about endonucleases cutting DNA at specific sites. We have said that these sites are generally palindromic, and we have said that some of these enzymes can generate sticky ends, meaning that they leave certain nucleotides exposed, single-stranded DNA exposed with nucleotides having ability to form complementary hydrogen bonds with a similar sequence. So what can we do with this? So here's the genetic engineering. Basically, genetic engineering means piecing two different DNAs together, stitching two different DNAs together. So what people do is they take a plasmid vector. What is a plasmid vector? Plasmid vectors are small circular DNA pieces that are present in bacteria. We have already talked about this. Bacteria has one large chromosome, which is a circular DNA molecule. In addition to that chromosome or DNA molecule, there are other smaller circular DNA molecules present in the bacteria. They also, of course, contain genetic information. These smaller DNA molecules are called the plasmids. So bacteria has a large chromosome and several plasmids generally in it. If we take a bacterial plasmid, isolate it, we take a foreign gene and we cut both of them with the same endonuclease or the same restriction enzyme. We will generate sticky ends since they have been cut with the same enzymes, and these sequences are palindromic. So these sequences can bind not only among themselves, but these sequences can also bind the foreign DNA which has been cut with the same enzyme. So if you allow this reaction, you mix these two together with the enzyme, the probability is some of the times the plasmid or the vector the sticky sequences will fold on themselves and restore the original plasmid vector. But sometimes it may also allow the foreign DNA which has been cut with the same enzyme to insert here. When that happens, we have basically done genetic engineering. We have formed a new DNA molecule. So these plasmids can then be taken into and inserted, put back into a cell, a bacterial cell, where they will of course multiply, and we can have several uses for these. We will talk about the uses, but here I want to say that these plasmid vectors, they are put into a special type of bacterial cells which have been modified in such a way that they can accept these vectors or these plasmids. This was one of the ways people used to amplify DNA before the PCR was invented. They will take a sample of DNA of interest, they will cut it and piece it together in a vector or plasmid, put it back in the bacteria. When the bacteria grew, made many copies of themselves, so did the plasmid. They would extract the DNA and then cut the same DNA fragment out of them. It was long, laborious, tedious. Thank God PCR is here and we don't have to do this anymore. So now imagine this, that we have this plasmid or expression vector, we can call it now. We insert a specific gene in it and we put it in the bacteria. What can we do with it? You may have heard that people make insulin using biotechnology or genetic engineering. So we can do the same thing. We can, if we have insert the gene for insulin in the expression vector or the plasmid, put it back in the bacteria and allow the bacteria to grow. When the bacteria make messenger RNA from our gene, it will be translated into protein and we can harvest the protein from these bacteria. So here we are making these bacteria do work for us, make special protein which we need for our health. What are the other uses of cloned DNA? People also use the cloned DNA through the plasmid vectors to sequence initially. And also, not only they sequenced the DNA, they also were able to find the sequence of the protein, the sequence of amino acid in the protein that would result from that particular gene. It was also possible to use this technology and cause mutation, change one or two amino acids in a protein and see how it affects the function of the protein. So what is the role of a particular amino acid in the protein? Of course, I have already mentioned it was also used to mass produce different proteins. TPA is also one of them. Insulin, which is commonly used, a drug which is used by people who have diabetes. A certain type of diabetes, they use insulin. So biotechnology has had a major impact on the human life as we know it.